Good to be back here with you. We're working on a survey of microbiology by Tortora, an introduction. We've talked recently about bacteria that cause human disease, various eukaryotes, and now we're ready to jump into chapter 13 about viruses, disease-causing viruses. There's a little picture of some hepatitis A viruses that cause an acute liver infection. So viruses, as we've mentioned several times, and kind of been leading up to this for a while, mentioning them here and there, they're obligatory intracellular parasites, and that means that they can't do anything on their own. They're inert particles until they get inside of a host cell, and they don't have any organelles, they don't have any ribosomes for making proteins, they really can't do anything. The thing about them is they have a very defined host range, it's called, or ecological niche, or niche. Viruses have surface proteins that must attach to a receptor on their host cell or target cell, and then they enter the cell either by fusing with the plasma membrane or by receptor-mediated endocytosis. So that's how viruses get into their host cells, and so they don't infect randomly, they infect very specific types of host cells. They typically have some protein on the surface, which are antigenic, but some have especially long, pointy surface proteins called spikes. Not all viruses have spikes. Some viruses have a lipid bilayer on the outside that comes right from the cell membrane to the host as they exit, and that's called an envelope. Not all viruses have an envelope. What they do always have is a protein capsid around, protein coat or capsid around the DNA or RNA that makes up the genome with virus. Here's just a cool picture showing you a gigantous red blood cell, which incidentally is a pretty small cell on the scale of human body cells, and yet here's the size of a bacterium, how much smaller that is, and then look at the size of the viruses. They're extraordinarily tiny particles. Viruses weren't discovered until quite recently because of their tiny, tiny size. Nobody could see them in the microscope, and there's really no easy way to know that they're even there. So a lot of diseases that were frustrating because they had no known etiology turned out to be viral diseases. Ultimately, viruses were finally discovered, and ultramembranes were produced that could capture them by filtration and so forth. But a hint about viruses came along when it was shown that certain diseases could be recapitulated by using body fluids from someone with the disease that had been filtered to remove all bacteria and introduced into another host, and it would cause the disease. So clearly something was there that was too small to catch in the filter, and so that led to a search for something smaller finally discovered. So viruses, a virion is the active form of virus, a virus that can actually infect a host cell. It's got everything it needs, it's ready to go. There's got to be a genome, some genes, the nucleic acid, DNA or RNA. Viruses are very versatile in that way. Some have DNA, some have RNA, some have double-stranded, some have single-stranded, some have multiple copies. We won't worry too much about that, but just for interest's sake. There's always a glycoprotein coating around protecting the genome called the capsid. It can have an array of different shapes. Sometimes, again, there's a lipid bilayer around the outside called the envelope. There may or may not be spike proteins projecting from the surface. Here are some possible configurations of the capsid in terms of shapes. We'll look at polyhedral virus capsids, helical, and then we'll look at complex. So here's a very typical looking polyhedral virus. It's like a geometric volume container. It's got multiple flat surfaces, faces, and in this case you can see some spike proteins sticking out of this mastadenovirus. Pretty cool looking, and you can see the genome all tucked inside that capsid. Each protein monomer that assembles together to make the capsid is called a capsomer. And those proteins have to be synthesized in the host cell. There's another beautiful micrograph with a bunch of polyhedral viruses. Helical viruses are arranged of monomers that are formed into a tube. So each one of those monomers kind of forms a spiraling and elongating structure of a tube in which the genome is maybe found. And here's another picture of a helical virus. Ebola virus is a very terrible disease-causing virus that is a helical virus as well. Influenza virus, helical virus, all wrapped up in an envelope. Herpes virus, example right there. Now, a complex virus is shown here. These so-called T-even phages that infect specifically bacteria are called complex viruses. They have a head where the genome is incorporated. They have a body, a sheath around this body, like a hypodermic needle. Then they have a bunch of legs or tail fibers, and so there's a lot of parts to them. They're quite interesting indeed. They, as we'll see, well, we'll wait and say anything more about that. How about naming of viruses? There are family names. That's as broad a category as there is when it comes to virus names. Then comes the genus name, and then the species name. Occasionally, viruses are named for subspecies, subgroups that are designated by different numbers. For example, herpes viruses. Human herpes viruses are called HHV, and there's HHV 1 through 8 that can cause human disease. So we'll be seeing all those, and we'll find out there's also common names for a lot of those, because a lot of the names of viruses were provided long before the more modern classification schemes came into vogue, and so we don't want to throw away those names because they're familiar to people. So we have herpes viridae, and that's the family, genus herpes virus, and then human herpes virus 1, 2, and 3. And there are, of course, as I said, 5 through 8 as well. We'll look at those later. Retroviruses, Retroviridae, with the genus lentivirus, and in that category, in that genus falls the human immunodeficiency viruses 1 and 2 that cause HIV and AIDS. So again, viruses have to enter inside of a host cell in order to quote-unquote live. They're not really living, they just have the right machinery to co-opt the protein-making equipment inside of a cell so that they can have their genome copied and all of their capsid proteins and various enzymes that specialty enzymes they often come with, can be recapitulated and packaged up into new virus particles. Bacteriophages infect bacteria, and when they do, they lyse the bacteria. So a way that they're often prepared is to grow a lawn of bacteria. It's just a confluent solid mass of bacteria growing on the entire surface of an agar plate, and then infect with bacteriophages. I actually mix those in first and let the bacteria grow up, and the phage will eventually mature and lyse a bunch of bacteria in a patch called a plaque. So there's a picture of a lawn of bacteria. This plate would look the same color as all those clear spots if it weren't for the bacteria growing there. And then those clear spots are plaques, places where the phage has infected the bacteria and burst them and lysed them all. Animal viruses can be grown in living animal cells or in embryonated eggs. So if you have a chicken egg that has started to develop a chicken embryo, you can take that egg, introduce viruses into it. The viruses will be able to grow in the cells of that egg embryo, and inside of that embryo, and produce lots and lots of viruses, and many viruses are manufactured that way. Alternatively, viruses can be grown in the laboratory in tissue culture, can grow immortalized cell lines. These are cell lines that are able to be grown indefinitely in culture, and then we can transduce them with viruses and collect virus particles from them. So here's a picture of an embryonated egg. You see the little chicken embryo there forming, and a bunch of different locations where you can inject different viruses. I'm not going to don't worry about all those names. There's different names for the different membranes that make up parts of this egg structure, and there's various places that have been successfully used to inject different types of viruses and get viruses to grow in the cells of the embryo and of the membranes. Here's a little picture illustrating the idea of taking some human tissue, digesting it with enzymes, and separating the cells, and you can grow those cells in tissue culture. Now, the primary cells, the ones come directly from the human tissue and can be grown in culture, but they're not very good for growing virus all the time because they can only double or divide a certain number of doublings, and then they age out and stop dividing, become senescent. But if you let primary cells sit in culture long enough, eventually a cell will acquire a mutation that allows it to continue growing without that aging out process, and now you have an immortal cell that can grow indefinitely. That's called a cell line. Some types of cancer cells can be isolated from human tumors and used as cell lines. Sometimes those cell lines are spontaneously arising cell lines in tissue culture from primary cells, and they are equally good for growing viruses. So this little pile of cells represents some transformed cells or some immortalized cells. They're just, authors are just trying to point out, they're not exactly the same as the primary cells were, because they have some genetic change that allow them to be immortal. Oftentimes, though, they're very, very similar in phenotype to the original cells, these immortalized cells, and so they make good models for studying the functions of these cells. But anyway, in this case, they make good cells for growing viruses because they're cheap to grow and they can grow indefinitely in the laboratory. When you transduce a cell with a virus in this sort of situation, as the viruses accumulate inside the host cell, the cell will look very, very different than it did when it was growing normally. The morphology or shape of the cell will be noticeably changed. That's called a cytopathic effect. So you can recognize viruses by the cytopathic effect they produce in cells. Another way you can identify viruses is by serological testing, using antibodies that have been produced against virus proteins in animals, say, and then use those antibodies to probe a tissue sample or a fluid sample from a human to see if the antibody sticks, and it would indicate the presence of some virus proteins. Or you could test for antibodies in a body fluid by using some antigens to see if it would stick to the antibodies, and the antigen may be a purified protein from a particular virus with a fluorescent tag on it or something like that will allow us to see the antibodies against the virus. And if someone has antibodies against the virus, it stands to reason that they have been infected with the virus. There's a number of different serological tests that we won't go into right now. Hemagglutination, for example, is a cool test, and we'll talk about that when we talk about applications of immunology. I don't know if it's still being done, but the major way that various strains of influenza were tested around the world every year to decide which ones to incorporate into the annual influenza vaccine was by hemagglutination inhibition assays. And the CDC was available, or the World Health Organization, throughout the world, so they could collect data on what the most common strains of influenza were around the world that year. Excuse me. Nucleic acids can be used to identify viruses. Restriction fragment length polymorphisms. I don't know if you remember, we talked about DNA fingerprinting. That's just a fancier name for that. Restriction fragments. A restriction enzyme, remember, is an endonuclease that can cut DNA at specific sites. We take the genome of an organism, we have a cell sample, a tissue sample, for example, and we digest it with some restriction endonucleases. We'll get a bunch of fragments. You run that by gel electrophoresis out and separate all the fragments by size. Particular fragments are diagnostic, sized fragments are diagnostic for the presence of certain viruses. So that's what's meant by restriction, fragment length, polymorphism, differences in the pattern of bands that's produced. And PCR finding. PCR, as you remember, is very, very sensitive because in PCR we amplify, double and double and double and double a target DNA sequence. And in this case, we would use primers that recognize a particular virus genome and take a human sample of someone who has disease and use primers against a certain virus that we suspect is implicated in the disease, and just amplify that thing to ad infinitum until we wind up with just billions and trillions of copies of the original DNA sequence if it's present, and then we can run that on a gel and identify it, or even sequence it and recognize the fragment that's specific for a certain virus. But remember, the primers for PCR are specific for a certain DNA sequence. So if we get a product, it has to be that particular target DNA that we have suspected, in other words, the virus. Here's a picture of cytopathic effect. Here are some most likely fibroblasts grown in culture. They grow like, they look like long, stretchy strand cells with maybe a couple of like forked, branchy parts in them. But over here are what they look like when they've been infected with viruses. And so they get all plump, and instead of being all long and stretched out, like they get all kind of rounded up off the surface of the plate. All right, let's talk about something different now. The lytic cycle of a virus, and particularly a bacteriophage virus and a lysogenic virus. We've mentioned this before. We'll just take one more look at lytic and lysogenic cycles. So in the lytic cycle, what are the steps? The virus has to attach to the target, the host cell. In this case, it's a phage. It's going to attach to a specific strain of bacteria, species of bacteria. And then it's going to penetrate. And in this case, it's a T-even phage, the complex phage that we looked at earlier. And what it's going to do is actually inject its DNA genome into the bacterial cell. So in this case, the virus doesn't actually enter the cell; it just injects its DNA right into the cell. That DNA then will activate synthesis of all the viral proteins to make new virus particles and copies of the virus genome. And then in the maturation phase, you'll have assembly of all the new phage particles, all their heads and bodies and tails, until we get a bunch of new viruses, and then they will eventually break the cell wall open and be released. Now, a lot of viruses come with specialty enzymes that help them either produce the new virus particles or, in this case, help them get out. So these phage have lysozyme genes, so it can produce an enzyme that breaks down the bacterial cell wall, weakens it, makes it fragile, and as the bacterium fills up with virus particles, eventually it bursts and release all those out. The release phase. When the virus infects, you put some virus into a population of bacteria, the number of free viruses drops to nothing as they bind onto the virus, onto the bacterial cells. That's what's meant by this number of virions. How many active virus particles are floating around in the solution? None for a moment, while this so-called eclipse period, while the viruses are all attached to the bacteria. Then they inject their genomes, and then the synthesis phase starts, and start producing all these biosynthesis phase, start producing all the virus proteins and genomes, and assembling viruses. Then we have a very large number of virions that are being released out from these host cells, and eventually there will be no more host cells left to produce virus, and the number will drop right back down again. So that's the lytic cycle. Here's the lytic cycle in cartoon picture form. Virus attaches to the bacterium, injects its genome, penetration, biosynthesis. You can see that we're going to be producing a whole bunch of capsid proteins and all the structural proteins of the virus, plus some genomes. The purple is the new genomes, and they're going to start to be assembled. That's the maturation stage, when we see some whole virions being put together with all those parts, and eventually there's going to be release, and they're going to escape from the host cell and infect more bacteria and form a plaque. Incidentally, it'll come up again in a bit, but I just wanted to point out that another enzyme that's present in the genome of this phage is one that can chop up the bacterial genome, so the bacteria isn't going to be making any of its own proteins anymore, no longer produce any messenger RNAs. The virus is going to be the sole driver of protein synthesis. So that's the lytic cycle. The lysogenic cycle, that involves incorporation of the phage genome into the chromosome of the bacterium. That's called a prophage. All right. So let's see, where shall we start here? So here's our phage injecting its genome, the penetration step. And in this case, the lysogenic cycle, we're going to be focusing on the right-hand side of this diagram. The left-hand side is the lytic cycle that we've already just been talking about. In this case, the genome that's been injected of the phage will circularize, and then it will become incorporated into the genome of the bacterium. So this little circle is supposed to represent the circular genome of the bacteria, even though it would be a very, very large, complex DNA molecule. That looks more like a plasmid, right? But it's the genome of the bacterium. And then whenever the bacteria divide, they end up copying the prophage right along with it. So now we have a large population of bacteria that have the phage DNA incorporated, and then the bacteria the whole population of bacteria may revert back to the lysogenic cycle, and the prophage will come back out again and take over the cell and start producing virus particles. So instead of having one phage infecting of one bacterium producing some phage particles, we have a very large population of bacteria, all of which can then revert to the lytic phase and produce a large amount of phage. So that's called the lysogenic phase. During the period of time when the cells are just dividing and there's no viruses being produced, that's a latency, a latent period in which nothing is really happening with respect to the virus. And finally, we'll talk about transduction and then special transduction. We've already talked about transduction before, and that's a situation where when phage are being biosynthesized and matured in a bacterium, their new virus genomes get incorporated into the virus particles, and they burst out and they infect new cells. But occasionally a fragment of the genomic DNA of the bacterium gets packaged up into a virion, and then when that virion injects its DNA into a recipient cell, it's injecting bacterial DNA. So brand new genes, brand new to this recipient cell, can come from this original bacterium, original host, can be inserted into this recipient cell, and then that can become incorporated into the genome. So now this bacterium has just received a brand new gene or genes that it didn't have before. So when we were talking about horizontal gene transfer, we talked about transduction, and that's just a review of transduction, the process of phage transferring genomic DNA from one bacterium to another. Specialized transduction is very, very similar. It's just that sometimes during the lysogenic phase, when a bacterium returns to lytic phase and the phage genome is freed up from the bacterial genome, it may not be a neat and tidy operation. There may be bacterial genes that are left attached to the phage genome and ultimately packaged up in a virion and then transferred to a target cell, another host cell. So that's another way, instead of by chopping up the genome and accidentally packaging up some bacterial genes, we have some bacterial genes that were attached to the phage genome when it was released from the chromosome of the bacterium and then transferred to another host cell during phage infection. All right. So that was really kind of a review, but anyway, there I go. Multiplication of animal viruses is not particularly different from phage and bacteria. The biggest difference is that animal viruses have to attach to the cell membrane by way of specific recognition of viral proteins by a receptor on the host cell. And then the virus has to get inside of the cell. It doesn't inject its DNA like a phage. It's going to get inside the cell either by inducing endocytosis. Once the virus binds the receptor, the cell can sense that on the surface and it will incorporate that virus into the cell. Or envelope viruses, the envelope of the virus can just fuse the plasma membrane, plunk that whole virus particle that's capsid with its genome right into the cytoplasm of the cell. Okay, so once the virus is inside, there's uncoating that happens. Got to get the capsid off of there because we need to free up the genomes in order to do biosynthesis. We have to have a step where we're producing copies of the nucleic acid and new proteins to make new virus particles. That's the biosynthesis stage. All those newly made proteins and the genomes can be assembled into mature virus particles and then released by two possible ways: either rupturing the cell, sort of like with the phage and the bacteria, or by budding. Some cells just leave by budding. And let's look at this picture. No, that's entry. Here's a picture of a cartoon drawing of a release phase of a helical virus by budding. The cell membrane is going to form a complete covering around the capsid of the virus called the envelope. All these blue dots represent viral proteins that were already inserted into the plasma membrane during the synthesis step, so that when the virus particle buds off and takes with it part of the cell membrane, it has virus spike proteins or virus recognition proteins right in the envelope. So that's the deal. Very similar indeed. This is just a beautiful picture of some viruses getting into a cell by endocytosis. You can see a vesicle forming around this virus. It's all attached to some receptors here, and soon it's going to go inside and uncode. Here's a fusion happening. Here you can see an envelope virus. You can see on the surface of the envelope are quite a few glycoproteins. Well, they're going to be left behind. The virus is going to fuse with the plasma membrane and end up inside the cell, and some of those excess glycoproteins were left at the surface as that virus went inside. And now you see uncoding happening as the virus capsid sort of dissolves away. There's another beautiful micrograph of budding of an envelope virus from a host cell. It's got that plasma membrane on the outside. A lentivirus, HIV viruses are released in that way. All right, join me again in a few minutes or I'll post in a few minutes, another part 2 of chapter 13, and that will conclude that chapter. Move right on to chapter 14 this afternoon.